So,
Okay.
good morning to all of you. Myself, Dr. Naresh Kumar Rangra, welcome you to the ISFCP sponsored dialogue series under the aegis of IQAC ISFCP cell. So, before starting the session, I would like to invite Dr. our worthy director, sir, Dr. GD Gupta, sir, to say some inauguration remarks.
So, thank you very much, Dr. Naresh Rangraji. So, first of all, I welcome from core of my heart from the ISF College of Pharmacy family as well as the IQAC. This is the dialogue series. So, today's expert, very dynamic and very experienced person, Dr. Sreedhara ji. And I welcome you, sir, from core of my heart in this
Thank
institute.
you.
And I feel that this types of the interaction, involvement, and contribution for the society, for the profession, I feel that it will be the highly beneficial to each and everybody. So, sir, this is the ISF College of Pharmacy. It was established in 1984. And right now we are
Mhm.
running the D Farm, D Farm, M Farm in 9 branches, Farm D, PB. And this institute has the also research approved center by the IKGPTU as well as the autonomous.
Very
So
good.
this institute in this region in North India, one of the premier institute in pharmacy. And this institute achieved 24th rank in NRIF, sir. So I hope that this types of the industrial contribution it will be the helpful to the each and everybody. And the purpose behind of this direct series, sir, to the interact with the some of the industry, academia, research person, so that we can give some of the fruitful and new things for the profession. Because I know that as and when we are the involved in the academia, then what is the going on in the industry and what is the recent advances in the industry sometimes will be the away from to me. As you know that Lackman is the very old book, third edition, even that we are going through it. While nowadays number of the digitalization, number of the equipment and machinery, they are the most probably is the changed. As we know that pharma is the now growing very fast, and we have feel that pharma 4.0, and now we are the preparing for the pharma 5.0 right now. While in the academia we are near about the 2.0 or 3.0, because digitalization, automation, this types of the things, obviously this is the very difficult in the academia, because the each and everything depend upon the what is the fee structure, government policy, and so many things. So for this purpose, this types of the dialect series give the recent advances and what is the development in the academia as well as the industry. And also I want some of the strong collaboration with the industry and academia, not only the paperwork only. I want in the actual form, so that placement of the students and the technology we can exchange to the each and other. So thank you very much,
Very
sir,
good.
for accepting our invitation. Welcome, sir. Over to Dr. Rangraji.
Okay. Thank you, Dr. GD, sir. So let me introduce to you today's speaker, Dr. Sreedhar Rao VOLETI. He is a founder and MD over CEO of INDRAS Private Limited. Sir is a alumnus of Andhra University Vishakhapatnam, postgraduate from Central University Hyderabad, and PhD from IISc Bangalore. He has earned two postdocs from USA, and Sir's research area is STEM portfolio management, entrepreneurship, policy making, and implementation for non-profit organizations, which connects industries, startups, and academia with the government organizations. Currently, Sir is passionately promoting the scientific entrepreneurship from young to seasoned researchers, students, and faculties to take up innovations in life sciences, ranging from agriculture, biotechnology, healthcare, IT, and pharma healthcare sectors. He's also actively promoting the IP technology transfer of assets of life sciences. As an entrepreneur, he has co-founded Theraxel Discoveries and later founded INDRAS Private Limited that consults and contracts and collaborates on drug design aspects with researchers of industry and academia. His continued passion lies in research ranges from drug design and discovery, currently focusing on therapeutic mitigation of COVID-19 and solid tumors in oncology. For his innovative technologies and research utilized by wide range researchers at Ranbaxy, he is a recipient of Global Appreciate Award by Ranbaxy for his outstanding contributions in R&D data digitization. He is also he has also established and promoted and made self-sustenance a scenario of the Aspire BioNest, a life sciences among incubator of UOH recognized as the best emerging bioincubator among the bionests of India. He has published more than 25 peer-reviewed articles in international and gave numerous lectures at domestic and international conferences. With this, I welcome Dr. Sreedhara Rao on his talk, Drug Repurposing in Finding Therapeutics for COVID-19. Over to you, sir.
Thank you so very much, Dr. Naresh, Dr. Rangra. I should call you Professor Rangra, right? Am I correcting spelling correct?
Sure, sir.
Very good. First of all, a very good morning to all the students who are participating in this webinar. And I'm sure majority of them are masters in pharmacy
Yes, sir. Most
and
of our
some PhD students, if at all anybody there in
yes,
that.
sir. Yes.
Very good. So I'll try to keep my talk simple enough and lucid enough to everybody can understand it. Okay.
Okay.
And if you have any questions, please stop. I will answer the questions either during the talk or at the end of the talk. Okay. No
Okay,
issues.
sir.
And I'm sure ISF College of Pharmacy, which has been very old in Punjab and especially in North India, very well known. And research is one of his repertoire. So with faculty joining there across India and everywhere, have I think nurtured research to some level by themselves and also encouraging students to do research in pharmacy. Let it be pharmacy has various departments. Okay. And one of the research component of pharmacy is to designing a drug or discovering a drug, let alone manufacturing or even processing or pharmacy pharmacognizance, etc. All these can be looked at. Okay, so I think today's focus is going to be the topic. What I'm going to choose is to on the discovery of new drugs, or how do you find drugs that can be reused for any target that we are looking at in the human healthcare. So this is a research that we carried out in the past 2 to 3 years as part of COVID-19, because which is a very pertinent and very difficult one, because lot many humans have actually contracted it, died because of it, and there is no medicine for that even today. And if I ask any student, what is the medicine that people have taken for COVID-19, I don't know which one will be the answer that they will give. And if there is an answer anybody give, I'll give a prize at the end of the talk, and I'll mail it to ISF College of Pharmacy. Okay. So with that, what I will do, I'll start sharing my presentation. Okay, give me 1 second. Select the window. And I hope everybody can see this full screen. Professor Rangra, if you can say yes, then I can proceed further.
Yes, sir. It's visible now.
Full screen, right? Very good.
Okay.
So, what is called COVID-19? It is called coronavirus. Okay. And 19 is the year when it actually made to public. Even the research has been continued for the past 15 to 20 years in that, only during 2019, because of the outbreak, the word COVID got out and touched the entire humanity with a scare. And of course, you can see the title is How We Mitigate COVID-19 by Drug Repurposing. So there are two ways of doing drug discovery. One is drug designing and drug discovery, which will take at least overall process of 12 years, which everybody knows. Otherwise, and of course, since COVID-19 is only for 2 years, 12 years of research cannot be possible. So we need to find some new and novel innovative ways in which we can identify new drugs. So that is possible only by researching the existing drugs, and that is called the drug repurposing. And my company's name is called INDRAS in short form, which stands for In Silico Discovery Research Academic Services Private Limited. What it basically does is, the INDRAS does is, in silico means computational. So we do drug discovery through computational first, and that research is applied to both industry and academia, and that is what I do in this company. And of course, I am the managing director and founder, and you have got my email IDs at the bottom. And if anybody wants to go through my website, that is there as www.indras.in. So with this introduction, I'll go to the next slide. How do we mitigate any disease? And before we go into any disease, it hits in time, which is very wrong. So in that way, the time management is what is very important. And in my opinion, time is considered to be health. So if you are in time, you will be healthy every time. And time is money. It has a lot of value. Wasting time is wasting money. And time is also knowledge. Knowledge comes over the period of time, and wastage of time will give you knowledge that is absolutely no useful. And last but not the least, time is valued, and we should value the time, whether it is wrong time, right time, doesn't matter. We should value it and we should learn from it. And in that way, I'll try to restrict my talk within 30 to 45 minutes without giving you too many details of research what I carried out. But this is the research that we carried out within 1 year. What we achieved, I will show you and I will demonstrate to you. And this was a project funded in the April of 2020. That is when actually the COVID-19 hit India. The actual COVID-19 started during the December of 2019, when it started in China. And then it started spreading across the globe by people flying around, meeting around, everything. And by the time it came to April and February, the entire Indian subcontinent got really affected. I'm not going to talk about statistics here, but February or March 20 onwards, we had the COVID-19 coming into India, and people were able to take solutions to it. And April 2020, both Government of India and a lot of private companies have actually started funding to help the humanity, because at that time, there is neither a drug nor vaccine, nothing was available. So every company started funding on their own to give rise to certain impetus to research and development, to find out new drugs or vaccines or any product that people can consume to stay safe. So INDRAS Private Limited, and there is another company called Reagen Innovations, which is the in vitro partner. We both wrote this project to Tech Mahindra for funding. And of course, you can see that entire process went through April of 2020 to March of 2021. It's a year-long project. And within 1 year, what we accomplished under the title called Mitigation of COVID-19 by Therapeutics. That means we need to identify therapeutics like drugs, that to existing drugs, which will work for them. And I'll give you the details in the next slide. And what is the requirement which Tech Mahindra put in for this proposal? One is, the main goal is to find therapeutics for COVID-19, and that is the mandate. And what is the financial constraint? It is only less than or 50 lakhs rupees for 1 year. We have to find within that much of money. And timeline given is less than or equal to 1 year. And what they want actually is they also want publications and patents, as many as one can develop. And the expected outcome at the end is return on investment. Return on investment means for 50 lakhs for 1 year, if they keep, what is it that they get out of it? That is the ROI. These are the mandates required. And from ideation to execution of this project, we have started. You can see my photograph on the left side. I'm Dr. VOLETI and from INDRAS, and Dr. Uday Saxena, who was a scientist who was a member of a discovery panel for atorvastatin, a drug used for cholesterol reduction from Pfizer. Later on, he became the chief scientific officer of Dr. Reddy's, and then he has started his own companies, both in US as well as in India, and became a millionaire. So he and we joined our hands. I proposed the computational part, and he proposed the in vitro part, experimentation part. Both can be accomplished within 1 year. And if you see the path of the progress, you can see that from April, if you see at the bottom of the screen, the initial work was done to find out antivirals, the existing antivirals, would they work for COVID-19? And then once that is set up, while that is work is been going on, the in vitro screen, it was set up by finding a peptide that is actually the active site for the COVID-19. I'll talk about the details of it. And that experimental setup was made. And then while we go for screening the entire drug bank of around 8,000 drugs, the antivirals will be screened in vitro. And while that is going on, what we do is we take some natural products of about 30,000 natural products across the globe, and we screen them. And when we give the results to them, what are the top 10 of all these antivirals or drug bank or natural products, they will screen all these products. And then by end of June or August of 2021, we will be able to publish some patents and publications. So this is the strategy that we adapted. And what is required for ideal profile for drug repurposing? Because a drug which is already existing is already in the market. So what all the things it has actually is, the properties are, there is a history of extensive human safety is known for the existing drug. So the desired property should be, yes, it should have a human safety. And it should be available both from the physical aspects as well as from the cost perspective globally. That is something which is required for repurposing drugs. And because it's already available and existing, most of the drugs cannot be patented. But is there a patentability scope for this repurposed drug for COVID-19 that has to be checkboxed? And these are all the broad requirements. And what all the specific requirements are, we know that COVID-19 binds to human ACE inhibitor. So it should not enter into human cells. So that is a requirement. So that has to be proved both from computationally as well as in vitro. It should also block inflammation. So it should not cause any pain during the process. And it should block cytokine storm. Because usually what happens is when virus enters into the body actually goes into the immune cells, will actually develop cytokines, which are immune cells. And these cells actually fight against the virus. Sometimes what happens is these cytokines also become their own enemies, and it creates a huge storm. That means there will be more production of cytokines will be there. And that is lethal to human health. We can't handle it. So that cytokine storm has to be blocked. And last but not the least, it should not cause any thrombosis. Thrombosis, clots, blood clots. So blood clot formation will lead to a lot of side effects, and heart function will be eventually blocked. So blocking the clot formation should be there. So every drug what we screen for COVID-19, we should have all these desired properties. And of course, this is a graphic that gives you all the details which I showed you earlier. and we just want to reduce the effect of it. So the COVID-19 drug will need to block the viral entry, should have the anti-inflammatory and antithrombic properties. These are the three things that must be clearly required. Otherwise, these therapeutics will not work for COVID-19. And why are we saying all these things? Simply because if it does not block the virus at the human ACE2, which we know that COVID-19, when first came, it blocked the airways and people were unable to breathe. So that is because there is an enzyme called human ACE2, angiotensin converting enzyme 2, that to which this virus binds and which actually carries oxygen eventually, which gets blocked. That's why people were dying because of lack of oxygen. And second thing was people were getting a lot of pain and a lot of inflammation, cytokine storm was coming up. That is because of the non-anti-inflammatory or severe inflammation that has been produced during this entire virus adding into the body. So that has to be reduced. And that inflammation, eventually what happens is causes blood clot formation in the veins, and this one, and those blockage has that clotting should be prevented. If clots are formed, then blood will not flow, and that will create a lot of issue. So that is because platelets aggregate, and then that causes the blood clotting. So usually blood thinners are given, but you can't give more blood thinners because they will become really toxic to the human body. So what do we know so far? At the time in March of 2020, there are people who are really running haywire to go to hospitals and ICUs, and doctors were clueless. So they were giving these following drugs: chloroquine, Caletra, remdesivir, favipiravir, etc. To all these are all the known antivirals that are available for other viruses, not for COVID-19. And these were actually for the use of that. Current use is given for what it has been developed. For example, chloroquine is actually developed for malaria, not for human ACE. Not for the COVID-19. Okay, and of course, all these things were tested. They were approved for usage, but they were approved to use for COVID-19 because nothing else was available. Okay. So idea is, how do we come up with new drugs? Now let's go a little deep into COVID-19. So COVID-19 virus has multiple proteins on its surface. It's called N protein, which is called nucleocapsid protein. S is for spike protein, which is largest, and you can see that in this graph top left, in that circle, you can see it is little longest one. It's like a flower coming out of the bud. And you have M protein, He protein, like hemoglobin, esterase, and envelope protein, and N protein, and S protein. These are very small ones, other than S protein. Sorry. The rest of the ones are small proteins. And these proteins are not available for drugging because they're just right on the surface, but they're not accessible. The only protein that is available is called spike protein. And this spike protein, which is called the main, it has got a huge binding site. Usually the mechanism in the pharmacy, I'm sure you learned that this is that a drug goes and attacks the active site and blocks its function or enhances its function for therapeutic benefit. And in this case, S protein has a huge binding site, and that is well studied earlier. With a lot of crystal structures are there, but there are no small molecules that can bind to it. They have developed actually protein therapeutics, peptides that would bind to it. But peptides cannot be used as therapeutics. So our strategy was actually to find alternative active sites within the spike protein where small molecules can go and bind to them and inactivate the spike protein, because spike protein is the one that binds to human ACE. So far, everybody is okay? People able to understand what I'm saying? Hello?
Yes, sir. Yes, sir.
Great. So, are there any questions? I presume no, and I'll continue.
No.
I presume no, and I'll continue, and people can ask me at the end or in between whenever they want. Okay, that's good. So, what does a spike protein contain? So now we have seen spike protein. You can see how it looks like. Okay, it is under the open reading frame 2 over F2, and it has got several. It has about 30,000 base pairs, and of course you can see that there are other sites that we can look at in the next slide. So it has got two domains. It has got about 1200 amino acids, 1273 to be precisely. And it's got two domains, S1 domain and S2 domain. S1 domains ends at 685. And from 686 on to 1273 will start around S2 domain. So you can see that both S1 domain and S2 domain are almost similar in size. Except S1 domain doesn't have any helices. S2 domain has most of the helices. And you see the RBD, which is the receptor binding domain, and that is in between the S1 domain and N2 domain. This receptor binding domain is the one that actually binds to the excuse me, to the ACE binding protein. So the conformation of S1 and S2 is in such a way that RBD is open to bind to the ACE. So if there is a way we can actually block S1 domain, then its conformation changes and it will not allow receptor binding domain to bind to the human ACE to reduce the virus binding to the human ACE binding site. Okay, and these are all the abbreviations that I've given, but don't worry too much about it. I want you to focus mostly on the RBD and the S1 domain. Okay. And now you can see that how does the main protease, that is the spike protein, and how spike protein RBD and the SARS-CoV will bind with each other. And you can see that on the right side, you can see that the RBD is circled in a golden color structure. And SARS-CoV-2 ACE2 is, the human ACE2 is actually shown in the blue color. So RBD is the one that we are looking at seriously. And if you look at the RBD closely, it is on the left side, you can see there are some beta sheets and helices are all colored in domain 1 and domain 2. And there is a catalytic dyad, the cysteine histidine catalytic dyad, that is the one that is very critical for ligand binding. And also, there is a substrate binding site located at the cleft between domain 1 and domain 2. That means in between that one, you can see that there is a red color circle and black color circle. That is the area where the substrates will bind. The substrate, when it binds to ACE2, that is the one that releases the inflammation into the human ACE2. Now, what we did was, you remember I told you that there is one main binding site called EMPRO site, which is well studied for peptides. So we can't use that. So our idea was actually to block alternative sites by finding them in the spike protein. So what we did was, computationally, there are various ways to find what are the alternative binding sites. So 6LU7 is the protein that we have taken, which is about 1120 amino acids. And it has a huge surface area of 1129 angstrom square and volume of 594 angstrom cube. That means only big molecules will fit in. Similarly, the EMPRO site is also as big as the 6LU7 and has a huge volume. That means actually even peptides can only fit in there. Small molecules, even though fit in, they won't be able to bind effectively. Then what we found was, we found actually 7 active sites, smaller ones, both in S1 and S2 domains. In S1 domain, we found 3 of them, which are designated by S11, S1-2, S1-3. And in S2, we found 4 of them, S21, S22, S23, and S24. Now you can see from the binding surface area column, S11 and S12 are around 600 to 650 angstrom square. So that is considered to be a small molecule's surface area. Small molecule like drug. Proteins will have larger than this. So, and of course its volume, the length, the breadth, and then the depth is about 150 angstroms cube. So that means its site score of 0.78 to 0.8 means it looks like a drug could fit in there. And if you look at S1-3, its binding area is so small, its volume is also like so small. So, and its site score is much lesser. So we clearly eliminated that. That's why it is highlighted as red color. Similarly, if you take S2 domain, which is not the ACE binding domain, we have seen in previous slide, the binding area is so large that anything can fit in. It's about 2,500 angstrom square. And volume will still remain the same. That means it is not depth-wise, it is not small, but shallow-wise, it looks like a Grand Canyon. Grand Canyon is is one of the natural wonders. It has a whole length, but it has very less depth. Even though its site score is higher than S11, but large molecules can only fit in there, not the small molecules. S23 and S24, like that of S13, they have absolutely no binding surface area and volumes are so small, so nothing can easily fit into that. So what we did was we considered S11, S12 only, and we ignored the rest three of them. So S22 and S23 were located due to location because they don't bind well. S13, S23, S24 ignored due to smaller in sizes. So graphically, this is how it looks like. So if you look at the proteasal empro site, that is where a molecule is been crystallized, that is been given there. It is really big in respect to volume and surface area, whereas the yellow color bordered S11 and S12 are small molecule can easily fit in, but larger molecules can't fit in there. Whereas S21 and S22 is on the towards the extreme right, which are not shaded. They are, you can see that there the cavity is really deep and it's really long. And larger molecule can only fit into that one. So we have considered S11 and S12 only for this research. And what all the things we studied in this one? Because these are small molecules, we studied three types of small molecules. The first milestone is computationally, we wanted to study all antivirus drugs, whether they fit into S11 or S12, and what would be their properties. And second set was we have taken around 8,000 compounds, which are already existing drugs in the world for various diseases. And we wanted to see whether how would they bind into COVID-19, S11 and S12 of spike protein. And third one is we've taken GRAS compounds. GRAS is nothing but generally regarded as safe. That means it includes natural products, nutraceuticals, or even C products, etc., etc. And there are about 30,000 of them. We want to screen all of them and see which one of them will bind to these two active sites. So the outcomes, if you look at, we try to prioritize 7 out of 22 the existing antivirals, and 10 from 8,000 compounds of existing repurposed drugs. And third one is 10 natural products prioritized from 30,000 natural products. So once we have 10 plus 10 plus 7, 27, we want to select the top 2 or 3 from each and then collect their biological properties in the in vitro. Now, how these drugs are fit into the entire natural product database and total drug bank database is given in this pie charts. And you can see that the 8,000 drugs are divided into around 30 to 35 classes of drugs. And then you can see 54% of them are mostly G protein coupled receptors. And that's all for other disorders. Okay. Similarly, the natural product database of molecules, there are 30,000 of them. You can see they are all created by molecular weight. And you can see that 1 to 200, 200 to 500, 500 to 600. So anything around 600 and beyond is what we wanted to avoid, because they become really large molecules. And anything between 1 to 200 is too small to bind to it, so we can't bind them. So we selected anywhere between 200 to 500, because which resembles mostly the natural drug molecules, natural molecules which are like small molecules. And also, if you look at PSA, which is called polar surface area, because this protein S11 and S12 are smaller, because they're small molecules, we don't want them to be anywhere beyond 100 to 150. It should be less than that. So what we did was we actually filtered anything which are highly hydrophobic, which will be having polar surface area of less than 50 angstroms, which is around 3600. And 10 to 150, 100 to 150 angstroms is around 17,388 compounds are there. So that's another one, which is the largest contingent. And anything bigger than 150 angstrom square, which means is a larger polar surface area molecule, we try to avoid them. So this is how we classified, and we try to connect the molecules for screening. Now we tried the antiviral drugs, the first milestone. And out of 27, we said, out of 22, we said the top 7, they're listed here. And of the top 7, the 3 molecules which are highlighted in the green have the highest delta G. Delta G is the binding energy, and highest ranking also, which both will give and interactions to the triads, etc. And we will be able to find the better interactions. Okay. So what we tried to find out was these 3 were the best ones which one can try for experimental evaluation. Otherwise, all 22 can be tested out, which will take a long time, and cost and energy and everything. As I told you in the first thing, time is value, time is also money. We want to reduce the money in all aspects. Similarly, we have calculated the same properties of these binding properties of these 3 molecules, these 7 molecules in the S12 also. The 2 active sites possible, so it can bind anywhere. We don't know where it binds, but it will bind there. And they also bind to Empro as a comparison. Okay. And if you look at, this is how the different conformations of these 3 molecules bind into S11, S12, and Empro sites. And you can see that there is more conformational consistency of these 3 molecules in the S11 and S12 sites, giving rise to more plausible antivirals might work towards COVID-19. Then, of course, this is how they look into the active sites. We don't want to worry about it, the interactions, etc. Then we went to the 8,000 compounds, and we prioritized top 10 of them, which are given in the green color, both in S11, S12, and Empro. And they were further, the third one is, of course, we've taken the natural products, and these natural products, top 10 of them, we gave it 5 of them, how they bind best. The best ones bind in the S11 and S12 and Empro. And what we found out in the drugs was there is a drug called ertugliflozin, if you can see at the bottom of the screen, the last one, which has the best binding energy and best rank score among all of them. So we have taken that, and this is a sodium glucose transport 2 inhibitor through kidneys in diabetes. This drug is considered to be the best one among all of its family members. Okay, there are about 10, 50, there are about 10 of them are there. Computationally, we have calculated which one is the best, and it turns out ertugliflozin is the best one. And right now it is not in India, but there is a drug called canagliflozin, which is bottom from the table, bottom top from the table, the second from the bottom. And of course, metformin is a drug which is commonly tested. We compared it. It's absolutely no use. So canagliflozin and ertugliflozin are 2 drugs that are supposed to be very good for binding into receptor binding site of COVID-19 to mitigate that one. Similarly, we have given so many rank scores and doc scores of various other final drugs in the three active sites, S11, S12, and S13, and EPRO. So that's how we have come up with drugs which are going to be useful for inflammation and COVID-19 mitigation. And we also compared that with antivirals, and we have taken some natural products, and we also compared that with this one. And just to prove from computational prioritization of 27 compounds, 8,000 compounds and 30,000 compounds to top 10 of in 3 of each, experiments became very easy. So what the in vitro collaborators did is they have taken the area where the ligands would bind, they've cloned the protein, entire protein of peptide of 30 to 40 amino acids, and hopefully that kind of bends into similar area, and then after the active site, and then they have taken these drugs at the bottom, you can see along the x-axis, and along the y-axis they calculate the percent inhibition of that particular protein. So that was done at 50 micromolar, 100 micromolar, and 200 micromolar. And they wanted to see at what concentration the 50% IC50 can be measured for its effective activity. And if you look at pretty much everything like etigliflozin, citravinib, idopamide, and thyroxine, lapatinib, which is an anti-cancer drug, all these things actually, even at 50%, 50 micromolar concentrations, they're very efficacious to block the viral entry of the human ACE2. We have done just to make sure that 50, 100, and 200, but even at 50, there is an 80% inhibition by etigliflozin, which considered to be very good. That proved our computational hypothesis. Then we have various other drugs. They wanted to see how the cells, that means human cells, will be affected by these existing drugs for cytotoxicity at 100 micromolar. And you can see that the control is at 500 core 2. And you can see etigliflozin and all these drugs are actually, even at the 100 micromolar concentration, they are not causing toxicity. That means your IC50 can go up to that level. So even at lower concentration, even at 50 micromolar, nothing is going to happen to lung cells to demonstrate the drug safety. And next one is cytokine secretion. And you can see that there is a LPS-induced cytokine storm formation study. In the control, when you induce LPS, lipopolysaccharide, you can see that the cytokine storms increases. And ertigliflozin at concentrations of 100 micromolar, we actually decreased it. And at lower concentration, it will decrease further. And lapatinib and everything are actually behaving exactly like that of a control. So that shows that these molecules can reduce the cytokine storm. And last but not the least is, how does the lung monocyte adhesion is carried out and this is done in 3D bioprinted vascular lung model. And this actually gives the cytokine storm as well as the anti-thrombosis process. And you can see etigliflozin does much better even at 100 micromolar with or without LPS, much higher than the control level. So is dasatinib, so is lapatinib, etc. So that means these drugs will function very well like the REF control. And last but not the least, the vascular lung model for thrombus formation. In the LPS, as soon as you give LPS, the thrombosis starts setting in. But as soon as you give etigliflozin, it actually reduces. And so is dasatinib and other drugs better than the controls. So that means all these molecules have done really well, as when I said in the first 2 slides, what are the desired properties and which molecules have got. Etigliflozin has got all ACEs for all these clot formation, cytokine storm formation, inflammation blocking or blocking the viral entry, etc. All these things have been done. And since the atigliflozin is patented for SGLT2 for diabetes, type 2 diabetes, and it was never reported that it will be useful for COVID-19, it can be patented. And both atigliflozin and canagliflozin, both the drugs are very good, are available globally, and cost-wise, they're acceptable. Okay, and they're available for extensive human safety. So atigliflozin found out to be a drug that can give safeness and efficacy to all the drugs, compared to all the drugs for COVID-19. And of course, and what is the prediction? This prediction was done. And of course, SGLT2 inhibitors actually did all anecdotal evidence in the clinical trials. And tyrosine kinase inhibitors like lapatinib, etc., also had all these three properties. And it is approved for COVID by FDA with this one. And homoharringtonin is an anti-inflammatory drug that has all these properties. It is in clinical trials for COVID-19. And if you look at the existing antivirals like chloroquine, etc., they can block the viral entry, but they don't cause any anti-inflammatory or antithrombotic, anticytokines. So chloroquine is not to be used for COVID-19. And this was later proved by doctors. Even though chloroquine was the first one to be used by doctors, later they said these are of no use. So in summary and conclusion of this entire research, what we did was we filtered about 40,000 compounds, 38,000 compounds of three classes: natural products, antivirals, and drugs, repurposed, both from molecular weight and polar surface area. And we actually calculated the rank score and the binding energies against the two new active sites that we found for COVID-19 spike protein. And second conclusion is none of the existing antivirals are found to be effective to mitigate COVID-19, even though the doctors earlier used them. But we realized and we found and we effectively proved through in vitro that it is of no use. And last but not the least, out of all the drugs that we have, 8,000 drugs for various therapeutic scenarios, like only three classes: diabetes with SGLT2 inhibitors, tyrosine kinase inhibitors for cancer, and some specific anti-inflammatory drugs like homoharringtonin were found to be effective binding to spike protein, mitigating the blocking of binding of COVID-19 virus to human ACE receptor. So with that, what happened? This all took 1 year for us to complete. And what happened afterwards actually is, this is the research that came in newspapers, what we have done. And of course, these are all various newspapers. At the last one, you can see everywhere this research came in. And what we did, we published about 4 papers in a journal called BioRx. It is free and the journals are already there, and I can share if you require. And we also wrote a mini review within 1 year in a journal. And we also did a patent, both Indian as well as international patent, on 13th of August 2021. And what is more gratifying was actually there is an award called Aegis Graham Bell Award that gives research, that gives the awards for outstanding research in current problems. So for COVID-19, they analyzed several of them, and we were one of the finalists among the rest of the ones in the innovation of life sciences for Tech Mahindra. Tech Mahindra is the one that funded us, so it was given to Tech Mahindra. So with that, I thank you all for your patient listening. Hopefully I was able to I'm in time. Yes. And I first thank ISF College of Pharmacy for giving me this opportunity to share the science and excitement for the relevant research that we carried out for COVID-19. Because within this 1.5 years or 2 years of 2020 March or April till 2021 June, where the COVID-19 maximum prevalence was there. And of course, everybody survived, including you all, and I congratulate you for that. Even I am also, I congratulate myself for survival to do this research by taking the right vaccines. But since there are no medicines were there, these were the things that we proposed to the in the literature where people could actually take it forward. Because of lack of funding and interest in time, we did not continue further. Otherwise, any of these molecules, if done in the in vivo or animal models and in humans clinical trials, if they turned out to be good, they could be used as drugs for COVID-19. And ISF College of Pharmacy, I have to thank for allowing me to present my science. And of course, the IQAC organizing committee, Professor Ganti and Professor Ranga, for asking me to give this lecture, and I share my immense thanks to them. And Team INDRAS is both myself and Dr. Shalini Saxena, who has done PhD from BITS Pilani. She is the one who has done the entire computational work. And Team Reagin Innovations, which is the in vitro team, which was led by Dr. Uday Saxena and his team members. And Tech Mahindra for initiative of this entire work, trust in having us to talk about this work, to do the work, and sharing the funding with us. And with that, I close my presentation and open to any questions. Thank you so very much. Appreciate it.
Thank you very much for this mind-opening session, sir. So, any questions from the audience?
First of all, I want to ask students, how many of you taken the vaccine for COVID-19? Just raise the hand, or you can show some emoticon, like you have that at the bottom of the screen. Send a reaction. How many of you taken either Covaxin or Covishield? Well, at least there is one. That includes even the faculty also. Anybody can raise.
So the students are raising.
Well, of course, some people have joined and some people have actually raised their hands. See, because drug discovery takes 12 years, it is very difficult to come up with a therapeutic in a short time, in a 1.5 year time of COVID-19. Well, vaccines are developed against a dead virus or dead protein of the virus, so it's very easy. So, only thing is, because once you have a vaccine and that is you inject into a horse or anywhere, you develop antibodies to it and then you raise them, it becomes much faster. That is the reason why world has come up with faster vaccines. But in reality, to accept even that one, also there are a lot of clinical trials are required. But knowing the importance of COVID-19 and the timeline and the fast variants of various alpha, beta, gamma, delta, etc., we can't have enough time. So people have to give it in one go. So vaccines were developed much faster and much quicker. That saved a human life. So with all due respect to that, that is the one that takes the major precedence. But eventually, there should be drugs too. And these drugs are the ones which will give rise to hope for developing new drugs. If you take diabetes, started with metformin, there are about like 50 drugs are there right now, 50 or 100 drugs are there for diabetes, working from various mechanisms. So that is how the knowledge gets transferred. Okay. So small molecules will be coming from antibodies. One can take it and then see which part of the antibody is yanking to COVID-19. And that interaction, we can actually model it, small size it, and only use the required ones. Okay. So that is the way the research has to be conducted. But congratulations all everybody who has taken the shots.
Sir, one more question I would like to ask you.
Yes, please.
Sir, actually, what is the success rate of this in silico drug designing technique in repurposing drugs for therapeutic potential?
It's a
In
great
your
question.
terms, yes.
No, I think it's a great question. There's no doubt. Okay. First of all, drug repurposing itself is a reusing of existing drugs for some other. See, they were developed for something and they're used for something else. So there itself, success is kind of limited. Then whatever we are doing is we have 8,000 drugs. Now, if you take 8,000 drugs, take a two-dimensional matrix or three-dimensional matrix. One column will be 8,000 drugs. Now, 8,000 drugs were developed for 8,000 targets, but human targets are about 50,000 targets are there. Now you take each experiment, in vitro experiment, and calculate one data point, like an IC50 at some X concentration, 10 micromolar. That means you got 8,000 by 100,000. That means 8 billion experiments you have to conduct. And you can imagine how much time and cost it takes. Now I can do the same thing in a computer, taking all these 8,000 and 100,000, and I can keep on do computational studies by docking and calculating the properties very quickly. And now the advent of computers is large, computational power is large, the space is large. You can do all 8,000 compounds in one go in one target in 1 minute. That means you need 100,000 minutes. So you divide the 100,000 minutes by number of hours and number of minutes. You can see how much time it takes for each molecule. Then I'll get a landscape first. I will eliminate all the unwanted experiments where the binding doesn't happen. So that itself is a time saving, cost saving, money saving, everything. And information is there. Now look, this molecule doesn't bond to the target, so don't do that experiment. So that's how it starts with it. And now you exchange that one, expand that one to 30,000 natural products, to 100,000 targets. Now you can see how much complexity the experiment has to be done. And experiments have to be done not only in human, right? They have to be first tested in animals, like mice, rat, and then dog. Now that is the third dimension to the entire two-dimensional matrix. That means in your lifetime, you will never be able to do these experiments easily. So the in silico, it may not give you a drug. Like since you asked the repurposing, I'll give you the repurposing aspect of it. With the repurposing, in silico will give you realistically, we brought down 30,000 natural products, 8,000 drugs, and 27 antivirals into 10 of them each within 3 months or 2 months of each. That I don't think any experiment can do that. How much like having a supercomputer will cost something. You have to have a super supercomputer like instrument to do all these things. You have 96 well plates and 124 well plates and everything. Now you put 100,000 well plates and 8,000 matrix and you do one each. You see how big of an instrument is required. That runs into billions. Now I am not even talking about millions. I'm talking about 50 lakhs rupees. 50 lakh rupees is nothing compared to a 100 crore worth instrument doing all these things, and then the time, cost, reagents, everything. Okay, so, and of course, to give you, I'm sure I'll give an exercise to students. They can go through Google and literature, and I also welcome faculty to do that. How many drug repurposing drugs are there in the market? Drugs are there which are repurposed. I'll give you many of them. I have got tons of examples. One of the best examples right now which is going on is an anti-diabetic drug which was actually developed for eye retinopathy is now used for diabetic foot. Originally, it was developed as eye drops for retinopathy. Now it is used for diabetic foot. The clinical trials are almost completed. Now that's going to come into the market. This is as of 2023 I'm talking about. There is a company called Nova Lead in Pune. They are doing it. Similar to that, there were many such drugs are there. Okay. So there is value. But the question is, if I take a drug repurposed drug and say, hey, this works for some other drug computationally. Do you believe it? No, we don't believe it. Because computationally, I can actually draw an apple, but you can't eat it unless apple is produced. Computationally, I can draw a pizza on a nice screen with high resolution, everything, but that can't fill the stomach unless somebody has to make it. So success depends upon what you envisage, what you realize, and what you make. Success is not one term. Success is combination of everything. Okay? So we should look
Thank you,
at
sir.
from a global perspective.
Thank you for this very well-mannered answer. You have explained so many things in a very simplified manner. So, with this, I would like to thank you, sir. On behalf of IQAC ISFCP, sir, I would like to extend a really hearty vote of thanks to our worthy speaker, Dr. Sreedhara, sir, who has spared time from his busiest schedule to grace this occasion. And today we had an opportunity to hear your thoughts on the repurposing drugs for COVID-19. And I am sure that this will be going to encourage us in our future research events also.
Hopefully.
Thank you, sir.
Thank you very much, and thank you very much for all the students who participated. And since there are no questions asked by any student, I'm sorry there is no prize. Otherwise, I thought I'll send some prize to your College of Pharmacy. And this is the last option. If anybody want to ask, please ask.
Anyone from the audience wants to ask students?
All right, let's not push them. But thank you so very much.
Okay, thank you, sir.
Good day. I'll sign off.